Welcome to Module 2, Learning About Medicines. In Module 1, we covered proteins and mRNAs, their structures, how they're made, and the roles they play in the body. In this module, we'll learn about what properties make for a classic medicine and how our options have expanded with recent technological advances so that we're now able to rethink what a medicine can be. Specifically, we'll be addressing the question, how can proteins and mRNAs be used as medicines? Welcome to Medicines, Small Molecules, and Proteins. Please take a moment to review the learning objectives for this lesson. Let's start with defining the term medicine. The legal definition of a medicine, medication, or simply drug is a substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease. Many of us take medicines every day in the form of pills. The active ingredients of pills are generally known as small molecules. Let's take a closer look at small molecule medicines. As their name implies, small molecules are much smaller than a protein or mRNA, more on the scale of a single amino acid or nucleotide building block. Therefore, many can easily cross cell membranes to work inside our cells without need of a special delivery system. Small molecule medicines have multiple advantages. First, most are able to be formulated into solid forms that are stable for long times at room temperature. Secondly, most can be taken by mouth, and once dissolved by the digestive system can go everywhere in the body, including, sometimes, the brain. These small molecule medicines include antibiotics such as penicillin and painkillers such as ibuprofen and acetylsalicylic acid, or aspirin. And the list goes on, from medicines that treat hay fever, nausea, high blood pressure, and high cholesterol, to those that treat psychiatric disorders like depression and schizophrenia. Nonetheless, there are some noteworthy limitations. In particular, current-day development of each new small molecule medicine generally involves an initial screen through tens of thousands of compounds, followed by years of medicinal chemistry to mold an initial hit into a potential medicine. Following that, there are years of preclinical and clinical studies to prove that it is both safe and effective. Unfortunately, most small molecules drop out somewhere along the development journey, due to either unanticipated toxicities or lack of efficacy. Because of the need to screen so many compounds and the high failure rate, small molecule drug development is both very time-consuming and very expensive. The way almost all small molecule medicines work is that they bind to a particular protein and change how that protein functions. Some small molecule medicines slow down or completely inhibit the target protein's function. Others do the opposite and increase the target protein's ability to function. Therefore, modulating the activity of proteins is a general mechanism by which we can treat ailments and disease. But proteins themselves can also be medicines. They now make up a key class of medicines known as therapeutic proteins or protein biologics. Almost all proteins currently in use as medicines do their work in the bloodstream. Notable examples include insulin, used to treat diabetes, blood clotting factors used to treat hemophilia, erythropoietin used to treat anemia, and monoclonal antibodies, which have many different uses ranging from warding off viral infections to treating cancer and arthritis. As a class, monoclonal antibodies have proven so efficacious that between 2015 and 2020, the U.S. Food and Drug Administration, the FDA, approved about 10 new monoclonal antibody medicines per year. Individual proteins from viruses and bacteria can also be used as medicines, in this case to prevent future infection. These are called protein subunit vaccines. Since they only constitute one small piece of the infectious agent, protein subunit vaccines are generally safe and cannot cause disease. Instead, they prime our immune systems to recognize and respond to that specific virus or bacteria in the future. Now, let's discuss some of the disadvantages of protein medicines. Despite their advantages, protein medicines also have notable limitations. For example, for the first several decades of its use as a medicine, insulin was purified from animals. However, animal insulin has a slightly different amino acid sequence compared to human insulin, and was therefore prone to causing allergic reactions in patients. Even more dire consequences were suffered by hemophiliacs. Prior to the development of recombinant clotting factors, hemophiliacs were treated with clotting factors purified from donated human blood. Contaminated blood products led to thousands of patients contracting hepatitis in the 1960s or AIDS in the 1980s. This was only corrected when appropriate screening procedures were implemented in 1969 for hepatitis and in 1985 for HIV. While some therapeutic proteins are still purified from natural sources, these days most are made using mammalian cell lines that have been genetically engineered to manufacture the protein of interest. A word commonly used to describe these cells, as well as the therapeutic proteins they produce, is recombinant, which loosely means a new combination. Recombinant cells are grown in huge stainless steel vats called bioreactors. Each bioreactor requires significant industrial infrastructure to keep the cells alive and free from contamination. So making therapeutic proteins is a very expensive endeavor. Further, because each therapeutic protein is unique, each one separately requires significant work to figure out how best to stabilize and formulate it so that it doesn't degrade during storage and ultimately functions as intended once injected into the body. Finally, most proteins can't easily enter cells, meaning that almost all therapeutic proteins are limited to jobs either outside of cells, for example in the bloodstream, or in the special compartments known as endosomes and lysosomes that cells use to take up nutrients from their environment. But more than 90% of human proteins reside either completely inside cells or are embedded within cell membranes. So if we tried to make and use these intracellular and membrane proteins as medicines, they wouldn't be able to get to the proper location that they need to function. This is where the idea of mRNA medicines comes in. Instead of supplying the human body with a pre-made protein, we could supply just the instructions for making that protein, that is, the mRNA. This would enable our bodies to manufacture our own therapeutic proteins. And because these therapeutic proteins would be made inside our own cells, any type of protein, intracellular, membrane-bound, or secreted, could become a medicine. It sounds so simple, but of course it's not. Our ability to use mRNA as a medicine has only recently become possible due to decades of hard work by both academics and industry researchers. In the later lessons of this module, we'll learn about the many barriers that had to be overcome to enable the creation of mRNA medicines. Welcome back. Before we learn about how mRNA can be used as a medicine, let's talk about some of the properties generally required of medicines so that we can have control over their effects and ensure their safety. Please take a moment to review the learning objectives for this lesson. An ideal medicine has 3 basic attributes. 1, its effect is dose-dependent. 2, its effect is time-limited. And 3, it can be dosed repeatedly. Both classic small molecule and protein medicines have all 3 of these properties. A limited duration of effect is particularly important because if one experiences side effects, you can simply stop taking the medicine. Let's check to see whether mRNA has the right features to be an ideal medicine. Unlike DNA, which serves as the permanent repository of your genetic information, both proteins and mRNAs are inherently transient. With very few exceptions, most proteins are under constant flux. They have limited lifetimes and must be regularly replenished. Likewise, the instructions for making them, the mRNAs, are constantly made and destroyed, only to be remade again. This enables your body to rapidly change the amounts of key proteins so that you can respond to changing conditions. To illustrate this, let's consider your circadian clock. This is the timer in your body that tells you when to be active and when to rest. The proteins responsible for the clock are aptly named clock proteins, and they appear and disappear with astounding regularity every 24 hours. Your body accomplishes this with a daily cycle of clock mRNA creation and destruction. The amount of protein produced at any one time is tightly tied to the amount of mRNA present at that time. Over and over, for every day of your life, Your cells get their daily dose of clock mRNAs, driving expression of clock proteins. Although all proteins and mRNAs are transient, each different protein and its corresponding mRNA has been tuned evolutionarily to have an optimal lifetime. Now, let's learn about a way to measure how long proteins and mRNAs stick around. A common measure used to describe how long a particular substance sticks around is its half-life. Half-life is the amount of time required for half of a substance to disappear after it has been made. Structural proteins tend to last the longest. The protein chrysalin in the lens of your eye has a half-life of more than 70 years. Once incorporated into the lens, chrysalin cannot be replaced and needs to last you your entire lifetime. But this is highly unusual. Antibodies are among the longest-lived proteins, but their half-lives are only 10 to 21 days. Remember that huge protein Titan? Its half-life is just 3 days. Shorter still are the half-lives of proteins that mediate metabolic reactions. Remember that metabolic chart from Module 1? An example from this class is the protein glucose 6-phosphatase, the one that's missing in von Gurke disease. Its half-life is somewhere between 8 and 48 hours. At the very short end are signaling proteins like the hormone relaxin, whose half-life is less than 10 minutes. So protein half-lives can span from minutes to decades. The median protein half-life is estimated to be about 30 hours, or just over a day. So what about mRNAs? How long do they last? mRNAs are not nearly as long-lived as proteins. The typical human mRNA has a half-life of just 5 hours. Of course, like proteins, mRNA half-lives do cover a range, with the shortest being just a few minutes and the longest being about 5 days. Importantly, no mRNA lasts for your entire life. They are short-lived by design so that your body can make the appropriate proteins in the appropriate amounts as your body requires them. Because, 1, mRNA is transient, 2, there is a dose dependency between an mRNA and the protein it encodes, and 3, an mRNA can be induced over and over again, mRNA has all 3 properties of a classic medicine. In addition to these, there are 3 more key properties of mRNA that make it extremely attractive as a new way of making medicines. We learned about the first of these in the previous lesson. That is, mRNA has the ability to direct the body to make any type of protein, not just proteins that do their job outside of cells. So mRNA medicines have the potential to meet many currently unmet medical needs. These range from rapid development of vaccines against new pathogens as quickly as they emerge, to stimulating the immune system to fight cancer, to treating patients with inborn metabolic errors by giving them back the ability to make the protein they're missing. The second feature is the small manufacturing infrastructure requirements for making mRNA medicines. Unlike recombinant proteins made by tissue culture cells grown in huge bioreactors, the process of making mRNA medicines has a much smaller manufacturing footprint. mRNA medicines are made using solely chemical, biochemical, and biophysical processes that do not require mammalian cell culture. The equipment for these processes is relatively small and can be mobile, making the manufacturing facility highly adaptable. Because the infrastructure and equipment required to make mRNA medicines are relatively modest, manufacturing sites can be easily set up in multiple locations around the world. And because the sequence of nucleotides that make up an mRNA is simply information, when mRNA drug designers have successfully created a new mRNA medicine, say a vaccine against a rapidly emerging pathogen, that mRNA sequence can be instantly delivered digitally to every manufacturing site for local synthesis and distribution. In the future, as technology improves, it might even be possible to miniaturize the entire manufacturing process to take place in a single benchtop machine. The third feature is that unlike small molecules and proteins, mRNA is an information-containing molecule. It is not itself the active medicine. mRNA is simply a recipe that tells the body how to make its own medicinal proteins. Once a drug maker knows how to make and formulate clinical-grade mRNA, and how to direct it to the desired cell type, for example, immune cells for vaccines or liver cells for a metabolic disorder therapeutic, they can simply swap out the previous mRNA sequence with a new one to create a new mRNA medicine of the same type, for example, a new vaccine or a new therapeutic. mRNA medicines are therefore a platform technology. According to Techipedia.com, a platform is a group of technologies that are used as a base upon which other applications, processes, or technologies are developed. These days, most of us have a platform technology in our pockets, our smartphones. The phone's hardware and operating system constitute the platform. This platform then enables rapid development of countless apps that we all use every day. In the case of mRNA medicines, the hardware is the manufacturing equipment used to make the mRNA medicines. The operating system consists of the processes and know-how for making mRNA medicines at various scales. Importantly, all mRNA medicines of a particular type use the same raw materials and manufacturing processes. Thus, the need for re-engineering to create a new medicine is minimal, and this can greatly accelerate the timelines to develop the apps, that is, the individual mRNA medicines. Yet another advantage of mRNA medicines is that they can be easily multiplexed, meaning that more than one mRNA can be delivered in a single medicine. This can provide the body with instructions to make multiple different proteins at the same time. Remember that some proteins act as multi-protein complexes. If a particular application requires synthesis of the entire complex, the mRNAs to encode each component of the complex can be delivered simultaneously. Alternatively, mRNA vaccines encoding proteins from different pathogens can be combined into a single shot so that you can be vaccinated against several different infectious agents at the same time. Now that we've learned about the potential advantages of mRNA medicines, in the next lesson we'll learn about the many challenges that had to be overcome to fulfill this promise. Welcome back. In the last two lessons, we learned about the properties of mRNA that make it possible to be used as a medicine. Importantly, mRNA medicines enable the human body to make its own medicinal proteins by tapping into the natural process of protein production. In this lesson, we'll learn about the challenges that had to be overcome to realize this potential. Please take a moment to review the learning objectives for this lesson. First, let's watch a video about how mRNA is made outside the body. The first challenge to overcome was making mRNA outside the body. Remember, in our bodies, mRNA is copied or transcribed from DNA in the nucleus of our cells. Making mRNA medicines, however, requires a DNA template outside of cells. This is accomplished by inserting a specially coded piece of DNA into bacteria, allowing these bacteria to reproduce and make many more copies of that DNA, and then carefully isolating and purifying the amplified DNA. From here, the process to synthesize mRNA molecules is purely biochemical, that is, cell-free. The DNA template is first cut with special molecular scissors to linearize it. This linear DNA is then incubated with nucleotides and a special RNA polymerase called T7 RNA polymerase. The polymerase strings together the nucleotides in the correct order to produce the mRNA. This reaction can be carried out on many different scales, from a very small amount for research purposes, to enough for one patient for a personalized medicine, to enough for tens or hundreds of patients to treat a rare disease, or on an incredibly large scale to make tens of millions of mRNA vaccine doses in a single batch. Once the mRNA has been purified, it goes through multiple quality control steps to ensure that the batch no longer contains any DNA or any leftover reaction materials, and that the mRNA itself is intact. Next comes formulation. Because our bodies impose multiple biological barriers that prevent it from staying intact until it reaches the target tissue, mRNA cannot be dosed as a medicine on its own. It is not currently possible to take an mRNA medicine by mouth because it will be seen as food and be immediately broken down and recycled. Instead, most mRNA medicines are injected into a muscle if it's a vaccine, or intravenously, that is IV, if it's a therapeutic. Other mRNA medicines are being developed that go straight to the nasal passages or lungs through an inhaler, and still more are in development to be applied directly to the skin. Within our own bodies, mRNA doesn't normally exist outside of cells. When naked mRNA appears in the bloodstream or lymphatic system, our bodies assume it's either debris from dead cells or evidence of infection by an RNA-based virus. Our biological fluids are full of custodial proteins that clean up dead cells and defend us against viral infections, and they rapidly destroy naked RNA. Further, since mRNA is normally made within the cells that need it, the molecule contains no wayfinders for where in the body to go. Therefore, mRNA alone has no means for tissue or cell selectivity. Finally, even if it was able to survive and find the desired cells, it would have no way to cross the cell membrane to get inside. Remember that mRNAs are highly negatively charged, so cannot easily cross cell membranes. For all of these reasons, mRNA medicines require a delivery vehicle, some sort of packaging material that serves three purposes. First, to protect it from degradation. Second, to direct it to the desired tissue and cell type. And third, to help it gain entry into the cell cytoplasm. Welcome back. Now let's learn about mRNA delivery vehicles. Although many different types of mRNA delivery vehicles are being tested, the most commonly used to date are lipid nanoparticles, or LNPs. Let's dig into the anatomy of an LNP and the function of each component. Please take a moment to review the learning objectives for this lesson. Let's watch a video to learn more about lipid nanoparticles. Here is a lipid nanoparticle, or LNP. Inside, we have our cargo, the mRNA. The rest of the LNP is made of lipids, a form of fat. Lipids form the membranes surrounding all the cells in your body. They are greasy or hydrophobic molecules, meaning that they don't mix well with water. Instead, they naturally form lipid bilayers like cell membranes or spheres. So LNPs are essentially just really tiny fat balls. How do we make sure the mRNA ends up inside the fat ball? In Module 1, we learned that mRNA is highly negatively charged from the phosphates in its sugar-phosphate backbone. Because opposite charges attract, one way to get a lipid to stick to mRNA is to make it always positively charged. However, lipids that are always positively charged are not well tolerated by the body. To overcome this challenge, researchers used a clever engineering trick to develop ionizable lipids. These lipids are positively charged under some conditions, but not others. These lipids will coat mRNA under acidic conditions, but then switch back to being uncharged under our physiological conditions near neutral pH. Cool, huh? The attraction between mRNA and the ionizable lipids works to make sure they wrap around their cargo, but it isn't strong enough to be stable in a vial or to survive long in the body. Another layer of packaging is needed for strength. This is composed of a double layer of a different type of lipid, a phospholipid, with cholesterol interspersed. Both the phospholipid bilayer and cholesterol are important components of your own cell membranes. In this cryo-electron microscope image of mRNA LNPs, you can clearly see the dark interior containing the mRNA and ionizable lipid, as well as the lipid bilayer membrane surrounding this core. Each LNP contains between 4 and 6 mRNA molecules. For cells to efficiently absorb them, LNPs need to be within a certain size range. The typical diameter of an mRNA LNP is around 80 to 100 nanometers. That's 1/1000th the width of a human hair. They're so tiny that each dose of an mRNA vaccine contains trillions of individual LNPs. But LNPs are soft materials. If they were allowed to touch one another while in storage, they would fuse together, making bigger and bigger fat balls as time goes by. So we have one final layer of packaging to prevent the LMPs from fusing with one another while they're in the vial. This layer is something called a surfactant. The most common surfactant currently in use is polyethylene glycol, or PEG for short. PEG is found in many other FDA-approved medicines, as well as over-the-counter products like cosmetics and toothpaste. When the PEG-coated LMPs bump into one another, the PEG keeps the lipid bilayers from touching, thereby preventing fusion. Once the mRNAs have been appropriately packaged to keep them stable during storage, they need to get to the right place in the body upon injection. You've probably heard of HDL and LDL. These are both examples of lipid transport complexes. They ferry fats from one place to another in our bloodstream. To the human body, LMPs look very much like lipid transport complexes. Different lipid transport complexes have affinity for specific tissues and cell types. Similarly, LMPs can be engineered to have affinity for particular tissues and cell types. Once the LMP has reached the target cell, how does the mRNA get inside? Cells take up LMPs by a process called endocytosis. This is how cells ingest material, such as lipid transport complexes, from the environment around them. The cell membrane engulfs the material, then buds inwardly to create a special membrane-bound compartment called an endosome. The final step in mRNA delivery is for the LMP to fuse with the endosomal membrane and release the mRNA into the cytoplasm, where it can interact with ribosomes and be translated into protein. Once the mRNA is released, what happens to the lipids? The phospholipids and cholesterol are normal parts of your own cell membranes, so are metabolized like any other lipids in your body. But don't worry, the amount of cholesterol administered in an mRNA medicine is minuscule compared to what's already in the human body. So taking an mRNA vaccine or therapeutic has no effect on overall cholesterol levels. Once in the body, the ionizable lipids and polyethylene glycol have been engineered to break down into smaller fragments that are rapidly eliminated by normal metabolic processes. All of these fragments are gone within a few days. Now that we have an appreciation for how mRNA medicines overcome barriers to delivery, we're next going to learn about another set of barriers that had to be overcome, the innate immune sentinels. Welcome back. In the previous lesson, we learned about lipid nanoparticles, or LNPs, and how they protect mRNA medicines from premature degradation and deliver them into cells. But one last major hurdle that had to be overcome to create effective mRNA medicines was the innate immune system. Please take a moment to review the learning objectives for this lesson. Now, when you hear the words immune system, what probably comes to mind are antibodies, which are proteins in the bloodstream that recognize and neutralize a particular pathogen. Antibodies are made by B cells, one of the key cell types that make up our adaptive immune system. The adaptive immune system is what vaccinations tap into to protect us from getting sick or severely ill from a specific pathogen like SARS-CoV-2. The other key cell type in the adaptive immune system are T cells. A type of T cell called cytotoxic T cells recognize and eliminate any cells in our body that have been infected by a pathogen, thus preventing those infected cells from making more of that pathogen. While the adaptive immune system enables us to develop very strong defenses against specific infectious agents, it takes our bodies weeks to months to develop a new adaptive immune response and multiple days to gear it back up to fight a pathogen that we've been exposed to in the distant past. Thus, the adaptive immune system is too slow to be suitable as a first line of defense when we encounter an entirely new pathogen. This first line of defense is the job of the innate immune system. Rather than recognizing the unique features of a specific pathogen, the innate immune system recognizes features common to all pathogens of a specific type. For example, many bacteria contain a substance called lipopolysaccharide, or LPS, on their outer surfaces. Our innate immune system is always on the lookout for any LPS in our body. because this is one of the earliest warning signs of a bacterial invasion. Recognition of LPS sets off inflammatory alarm signals resulting in body aches and fever. Now, let's watch a video to learn more about our innate immune system sensors. A large number of viruses that infect humans have instruction sets in the form of RNA. Common examples are coronaviruses, flu, and polio. These viruses enter cells by endocytosis and then release their RNAs from the endosomes into the cytoplasm. But this process is not 100% efficient, meaning that some viral RNA gets released inside the endosome. Our own mRNAs originate in the nucleus and stay in the cytoplasm, so are not normally found inside endosomes. So our innate immune system has special protein sensors that are constantly on the lookout for RNA inside endosomes. When these sensors detect RNA inside endosomes, the presumption is that it must be dangerous. The RNA sensors set off an alarm signal, and proteins called interferons and cytokines are released, causing local inflammation and sometimes fever. Remember from the last lesson that LNPs also use endocytosis to deliver their mRNA cargoes into cells. Like viruses, release of LNP-encapsulated mRNAs into the cytoplasm is not 100% efficient, with some fraction of the mRNA being released inside the endosome. Thus, LNPs containing mRNA can appear to our innate immune system to be a potential RNA virus. To make effective mRNA medicines, it was therefore crucial to learn how to slip by the innate immune sensors in the endosomes. The first step to overcoming this hurdle was understanding exactly what molecular features the sensors detect. Work published in the early 2000s demonstrated that the feature recognized by some sensors is the RNA nucleotide U. When U binds to these sensors, the sensors get activated and start sending out danger signals. Soon after that discovery, further research showed that using modified versions of U, as long as they don't interfere with translation by the ribosome, can prevent the sensors from recognizing RNA in endosomes. It turns out that although A, C, G, and U are the most common RNA nucleotides, our cells also contain many modified versions of these. In fact, over 100 different modified nucleotides are known to exist in nature. One that is normally present in the human body is 1-methyl-pseudouridine, or 1-MY for short. If U is replaced with 1-methyl-pseudo-U as the nucleotide in the transcription reaction, T7 RNA polymerase will produce mRNA with 1-methyl-pseudo-U throughout. These 1-methyl-pseudo-U mRNAs are able to evade the innate immune sensors that search for viral RNAs in endosomes. But even if an mRNA molecule is able to avoid activating the innate immune sensors that detect single-stranded RNA inside endosomes, there is yet another defense system to avoid. This second defense system is on the lookout for long stretches of double-stranded RNA. Like the sensors we've already discussed, this second set of sensors evolved to protect us from viruses. In order to make new copies of themselves, all RNA viruses have to go through a phase where their RNA is double-stranded. This feature is not present in our own mRNAs, which are largely single-stranded. Thus, a second set of innate immune sensors patrols both endosomes and the cytoplasm, looking for long stretches of perfectly base-paired or double-stranded RNA. Again, when these sensors are activated, they set off alarm signals, causing the release of interferons and cytokines. Unfortunately, the traditional process of transcribing mRNA using T7 RNA polymerase also generates a small amount of double-stranded RNA byproducts. To make an effective mRNA medicine, these double-stranded RNA byproducts need to be kept to a minimum. Fortunately, mRNA drug makers have now found very efficient ways to decrease the amount of double-stranded RNA byproducts both by changing the transcription reaction conditions and by implementing strict purification protocols. Understanding all the ways the innate immune system detects potential invasion by RNA viruses has enabled mRNA medicine makers to find creative solutions to avoid setting off the innate immune alarm systems. Combining these solutions with efficient mRNA delivery systems, such as lipid nanoparticles, has opened the door to the development of many new mRNA medicines. The various ways we can use mRNA medicines will be the topic of the next module. Welcome back. In this module, we will explore the different types of mRNA medicines and their potential applications for preventing and treating disease. We will discuss how mRNA medicines can be used as vaccines to prevent disease or as therapies to treat existing disease. We will also delve into the potential of mRNA medicines to treat genetic disorders, as well as create an entirely new class of regenerative medicines. This module will provide you with an overview of the exciting potential of mRNA medicines to address a broad spectrum of human disorders. Welcome to prophylactic mRNA vaccines. Now that we've learned about the attributes of mRNAs that make them suitable as medicines and all of the challenges that had to be overcome to realize this potential, we're finally ready to dig into the types of disorders that mRNA medicines can either prevent or treat. Please take a moment to review the learning objectives for this lesson. First, let's talk about vaccines, specifically vaccines meant to prevent disease. This type of vaccine is known as a prophylactic vaccine. We'll start here because the very first mRNA medicines to be approved and widely administered were the mRNA vaccines against SARS-CoV-2. Until very recently, the traditional method of vaccination was to expose the recipient to a small amount of a pathogen that's been inactivated, or an active but weakened, that is, attenuated pathogen. Pathogens may be viruses or bacteria. The polio and typhus vaccines are good examples of inactivated pathogen vaccines, while the MMR vaccine, measles, mumps, and rubella, is an example of an attenuated pathogen vaccine. The safety of an inactivated pathogen vaccine depends on the pathogen being thoroughly inactivated or killed prior to administration. If the pathogen is not completely killed, the vaccine can actually cause disease. In an infamous case in 1955, known as the Cutter incident, Cutter Laboratories released a batch of poliovirus that they had failed to completely inactivate. When it was used for vaccination, it caused more than 40,000 cases of polio, lifelong paralysis for 200 people, and 10 deaths. Similarly, patients with weakened immune systems, or who take an immunosuppressant because they've had an organ transplant, are often advised against taking attenuated pathogen vaccines. This is because their weaker immune systems may not be able to control any type of infection, even by a weakened pathogen. The way all vaccines work is to expose our immune systems to a new infectious agent in a relatively safe way so that we can develop an adaptive immune response. The first step in building an immune response involves specialized white blood cells called antigen-presenting cells, or APCs. Their job is to constantly be on the lookout for anything unusual, ingest potential pathogens, and display proteins or fragments of proteins from it on their own cell surface. The proteins or bits of protein they display are called antigens, gen for generate, and anti for against. Therefore, an antigen is something that generates an immune response against a pathogen. Antigen-presenting cells travel back and forth between the tissues they patrol and nearby draining lymph nodes. When they arrive in a lymph node, APCs present any antigens they've picked up on patrol to immature B and T cells. If one of these B or T cells is able to recognize a presented antigen, it will develop into a mature B or T cell. Mature B cells secrete proteins called antibodies that can bind to and neutralize the pathogen. Cytotoxic T cells, on the other hand, seek out the source of the infection and eliminate any infected cells before they can be exploited to produce more of the pathogen. mRNA vaccines work in a similar but different way. With traditional vaccines, APCs ingest the entire pathogen. With mRNA vaccines, the pathogen is replaced by LNPs. The APCs ingest the LNPs carrying mRNA instructions for how to manufacture one or more pathogen proteins, then use their own ribosomes to make those proteins for surface display. Previously, we learned that mRNA is inherently transient. So once it's inside the antigen-presenting cells, it lasts just a short time. In animal studies, mRNAs formulated with the same LNP used for Moderna's SARS-CoV-2 vaccine were undetectable after just 3 days. Similarly, the antigenic proteins are only present for a limited time as well. Although a single exposure to a strong antigen can be sufficient to generate an initial adaptive immune response, a second and even third exposure often results in stronger and more durable responses. Like most traditional vaccines, optimal protection from an mRNA vaccine requires both a first or prime dose and one or more subsequent booster doses given weeks to months later. In the next lesson, we'll learn more about some special features of mRNA vaccines. Welcome back. In the previous lesson, we learned how vaccines work in general, and specifically how mRNA vaccines work. In this lesson, we'll learn about some other attributes of mRNA vaccines that make them so promising for future vaccine development. We'll also learn about some current limitations requiring additional research. Please take a moment to review the learning objectives for this lesson. One beneficial feature of mRNA vaccines is that they can be readily multiplexed. This means that instead of just delivering a single mRNA sequence supplying the instructions to make only one protein, several different mRNAs can be combined and delivered in a single vaccine. Multiplexing is especially useful when the antigen is a multi-protein complex. In the case of cytomegalovirus, CMV, one key antigen is a complex of 5 different protein subunits. Combining all 5 mRNAs, each encoding the instructions to make one of the individual subunits, into a single vaccine enables antigen-presenting cells to produce all 5 proteins simultaneously, assemble them properly, and efficiently display the complete complex on their outer surface. This multi-protein complex generates a much more robust immune response than if only one subunit were displayed. In addition to the 5 mRNAs encoding the multi-protein complex, Moderna's CMV vaccine contains a 6th mRNA encoding the CMV-GB protein. This protein serves as an additional antigen, enabling an even more robust immune response against cytomegalovirus. Similarly, multiplexing mRNAs into a single vaccine also makes it possible to create combination vaccines against multiple infectious agents. You're likely to remember that the SARS-CoV-2 virus evolved over time to produce different variants that also spread globally. In response, new bivalent mRNA vaccines containing 2 different mRNA sequences were developed to protect against both the original Wuhan strain of SARS-CoV-2 and a later emerging variant. In the future, mRNA vaccine makers envision developing combination vaccines against multiple different viruses at once, such as a combination respiratory vaccine against respiratory syncytial virus, also known as RSV, SARS-CoV-2, and seasonal flu. With combination or multiplexed vaccines, you might be wondering how drug makers manage to ensure that each LNP contains all the different mRNAs. Actually, this isn't necessary. Each mRNA sequence has its own potency, which is a measure of strength or power. In other words, how many mRNA molecules are needed to create enough protein to elicit an appropriate immune response. Because some antigens are more immunogenic than others, different antigens require different amounts of mRNA to elicit the desired immune response. For most multiplexed vaccines, the different mRNAs are mixed together at a predetermined ratio based on their individual potencies, and then loaded randomly into lipid nanoparticles. Because the LNPs are so small, each one can only hold a few mRNA molecules. But a single vaccine injection contains trillions of individual LNPs, and each antigen-presenting cell likely takes up tens to hundreds of these individual lipid nanoparticles. As a result, all of the different mRNA molecules are efficiently delivered regardless of which LNP brought them there. In theory, there's no limit to the number of different mRNAs that can be multiplexed. mRNA vaccines are limited, however, by the maximum tolerated dose, or MTD, of the LNP. That is, the largest amount of LNP that can be dosed without causing severe side effects or reactions. As the number of different mRNA sequences increases, the amount of each individual mRNA must decrease to stay under the maximum tolerated LNP dose. The limitation to the number of mRNAs that can be included in a single vaccine, therefore, is not how many different mRNAs can be packaged into a single LNP particle, but how much of each individual mRNA is needed to elicit the appropriate immune response. For this reason, creating multiplexed or combination vaccines requires significant research and optimization to determine the right proportion of mRNA sequences to get an appropriate immune response to every antigen. Now, let's talk about the resources and time required to develop vaccines. In this section, we'll compare the traditional process for making annual flu vaccines to the process for making mRNA vaccines. Most current-day flu vaccines, for example, are made from flu virus grown in fertilized chicken eggs, with each egg providing only a single vaccine dose. That means more than 100 million eggs are required every year just to make enough vaccine for the U.S. alone. Imagine how many chickens are required to lay 100 million eggs, and then how much space is needed to raise the chickens and store the eggs. The space where the eggs are kept also has to be carefully environmentally controlled for temperature and humidity to keep the eggs alive. That's a lot of animals, space, and energy annually. Other traditional vaccines are created from pathogens grown in large bioreactors using various types of tissue culture cells. These might be mammalian or insect cells. In contrast to current flu vaccines, 100 million doses of an mRNA vaccine require no chickens or eggs. 100 million doses of an mRNA vaccine can be made in 10 to 20 large-scale biochemical reactions using equipment no bigger than a large refrigerator. So mRNA vaccines have much lower resource requirements, and importantly, no animals or animal products are needed to make mRNA vaccines. Another advantage of mRNA vaccines compared to flu vaccines grown in chicken eggs is that mRNA vaccines can be produced very quickly. Moderna's original SARS-CoV-2 vaccine was manufactured and quality controlled in just 42 days, whereas producing a new flu vaccine from chicken eggs takes 5 to 7 months. Unfortunately, because flu mutates so rapidly, it's possible for the genetic makeup of circulating virus variants to drift substantially between the time vaccine manufacturing begins and when the vaccine is ready to go into people's arms. Further, because the flu vaccine is made from live viruses grown in eggs, there can also be genetic drift happening in the eggs themselves. These twin drifts contribute to lower vaccine efficacy. Dependent on the year, current flu vaccines vary from 60 to just under 40%. In comparison, there's no possibility of genetic drift in the mRNA vaccine manufacturing process. Combined with their much shorter manufacturing times, mRNA vaccines are likely to better match circulating viral variants at the time of administration. Therefore, mRNA vaccines against annual flu and other fast-mutating seasonal viruses could ultimately result in increased effectiveness compared to the current standard. Finally, the relatively small infrastructure requirements for making mRNA vaccines means that manufacturing sites can be distributed throughout the world. For most medicines, the production process and infrastructure requirements are highly specific to that individual medicine. Therefore, they tend to be produced in just a few centralized facilities and then distributed from there. In contrast, an mRNA sequence is simply a string of letters that can be stored digitally and sent anywhere in the world instantaneously. Thus, once researchers have determined an optimal mRNA sequence for a particular purpose, it can be sent out to a network of small manufacturing facilities for local manufacturing and distribution. Because all mRNA molecules are made of just 4 building blocks, the materials required to produce different mRNAs are the same regardless of their final sequence. Similarly, the ingredients and processes for making lipid nanoparticles is the same regardless of the mRNA sequence or sequences being encapsulated. This makes it very simple to replicate the entire process for local production and distribution. In the future, by enabling local public health authorities to quickly respond to new outbreaks, this could help decrease the spread of novel infectious agents locally, and therefore the threat of another worldwide pandemic. Manufacturing facilities for mRNA vaccines now exist on every continent except Antarctica. There are also efforts underway to miniaturize the entire manufacturing process so that it can fit into a single cargo container ready to be shipped anywhere in the world at a moment's notice. Future innovations might even enable mRNA vaccines to be made on the benchtop. In contrast to their many advantages, mRNA vaccines do currently have some notable disadvantages. One is the relative fragility of mRNA. You may remember that the original SARS-CoV-2 vaccines had to be shipped and stored at ultra-cold temperatures and administered very soon after being allowed to thaw. This fragility is partly due to RNA's chemical structure. It has a tendency to break into pieces. The rate at which this happens is highly temperature dependent, so degradation of intact vaccine is significantly slower when it's frozen. Given the severity of the SARS-CoV-2 pandemic and the lack of other alternatives, these strict temperature conditions were tolerated at that time. But maintaining a functional cold chain is very expensive and generally limited to more affluent and populated regions. Therefore, a strict cold chain requirement is not desirable under normal circumstances. The good news is that in the time since the first SARS-CoV-2 vaccines were distributed, mRNA vaccine manufacturers have already made tremendous progress in slowing down the chemical reactions that inactivate mRNA vaccines. For example, as of the recording of this course, mid-2024, Moderna's updated versions of SARS-CoV-2 vaccines can be stored for up to a month in the refrigerator. And there are other ways to preserve mRNA vaccine potency. For example, they can be lyophilized, which means the vaccine is dehydrated and stored as a dry powder. This powder can then be reconstituted with liquid immediately prior to administration. Alternatively, researchers are investigating the possibility of incorporating mRNA vaccines into solid microneedle patches. Microneedle patches could potentially be stored at ambient temperature, would be easy to transport anywhere in the world, and could be administered simply by applying the patch to the skin like an adhesive bandage. An additional advantage of microneedles is that they only penetrate into the dermis layer of the skin, not the underlying muscle. Our skin is the primary barrier that prevents entry of infectious agents. Accordingly, the dermis has even more antigen-presenting cells, called Langerhans cells, than do skeletal muscles. So although intramuscular injection is a very common means for vaccine administration, intradermal administration can generate even stronger responses with smaller doses. Similarly, the greatest protection against pathogens that enter the body through a mucosal pathway, such as the respiratory system, is achieved by delivering the vaccine to the relevant mucosal lining. This stimulates a much stronger mucosal immunity. So in addition to microneedles, numerous efforts are now underway to develop mRNA vaccines that can be delivered to the respiratory or other mucosal systems. Undoubtedly, now that the first mRNA vaccines have proven safe and effective, the next few years will see rapid advancement in mRNA vaccine technologies, both to make them more stable and to increase their effectiveness against a broad range of infectious agents. With that thought, our discussion of prophylactic mRNA vaccines has come to an end. In the next lesson, we'll turn our attention to mRNA medicines being developed to treat disease instead of just prevent it. Welcome back. In the previous two lessons, we focused on using mRNA medicines as vaccines to prevent disease. We'll now turn our attention to other types of mRNA medicines, medicines with the potential to treat existing disease. These medicines are known as mRNA therapeutics. Please take a moment to review the learning objectives for this lesson. Because mRNA therapeutics are so new, as of the creation of this course in mid-2024, no mRNA therapeutics have completed all of the clinical trial phases required for final approval by government regulatory agencies. Nonetheless, because of their potential to fulfill so many critical unmet medical needs, there are a multitude of mRNA therapeutics at various stages of development, with many already undergoing clinical testing. Let's first learn about how mRNA medicines might be used to treat cancer. Cancer happens when a particular cell in the body acquires enough mutations in its genomic DNA that it no longer responds to the normal signals that keep its growth in check. The cell begins to divide and multiply inappropriately, and a tumor starts to grow. Additional mutations are acquired as the tumor cells divide, enabling some of them to invade nearby tissues. Eventually, if left untreated, parts of the original tumor may start to migrate to more distant sites in the body, resulting in metastatic cancer. One of the most significant advances leading to new treatments in cancer in recent years was the realization that cytotoxic T cells, the same white blood cells that recognize and eliminate viral infected cells, also work to protect us from cancer. That is, in addition to protecting us from infectious disease, cytotoxic T cells are constantly patrolling our bodies to find and eliminate our own cells that may become cancerous. How do they do this? With the exception of red blood cells, every cell in your body displays on its outer surface around 100,000 copies of a protein called Major Histocompatibility Complex Class I, also known as MHC1. The job of these membrane-bound MHC1 proteins is to showcase fragments of all the different proteins that are made inside that cell. In a way, MHC1 acts like a badge, and the protein fragments it displays are the ID cards proving that they belong in the body and that they have a legitimate job to do. Cytotoxic T cells constantly monitor these protein fragment ID cards, looking for fragments from non-self proteins. A non-self protein could be a protein from an invading pathogen, a self protein that has become mutated and is on its way toward cancer, or any protein not normally present in the body. If a cytotoxic T cell finds evidence of non-self proteins, the cell presenting that information is targeted for elimination. That's why they're called cytotoxic T cells. Cyto means cell and toxic means poisonous. Cytotoxic T cells eliminate our own cells when they're producing something unusual. Although the number of specific or consequential mutations required to develop cancer is between 1 and 10, most cancers contain more than 10,000 so-called non-germline mutations. These mutations arise due to errors during DNA replication and cell division. The vast majority of these non-germline mutations aren't actually involved in cancer development and progression. But some of these inconsequential mutations can be used by the immune system to recognize and eliminate the cancer cells if they cause an entirely new protein to be made or change the amino acid sequence of a normal protein. These mutant proteins are unique to that cancer. They are called neoantigens because they are both new and now recognized as non-self. When fragments of a neoantigen are displayed by MHC1, their ID card says, "I don't belong." Researchers are now developing and testing individualized neoantigen therapies, or INTs, also known as personalized cancer vaccines, to take advantage of non-germline mutations and train the immune system to recognize an individual's unique cancer. An individualized neoantigen therapy is similar to a prophylactic vaccine in that it activates the body's adaptive immune system to respond to a specific threat. But in this case, instead of a foreign pathogen, the threat comes from cancer cells already in the body. The first step in creating an individualized neoantigen therapy is to map all the mutations present in that individual's specific cancer and not in their normal tissues. That is, identify all potential neoantigens present in the cancer. This is done by performing deep genome sequencing on both a tumor biopsy and a blood sample. Advanced bioinformatic analyses are then able to identify which cancer cell mutations will produce a neoantigen and predict which ones are most likely to show up as an ID card on the MHC1 badge. With those peptides identified, it's possible to design one or more mRNAs encoding them. Often, the mRNA provides the instructions for a single long polypeptide that will be chopped up into smaller pieces for display by antigen-presenting cells. After manufacturing the mRNA, formulating it into an appropriate lipid nanoparticle, and performing all of the necessary safety and quality control testing, the therapy is ready for use. Remarkably, the entire process from obtaining the tumor and blood samples to delivering the finished medicine back to the clinical team can be done in under a month and a half. To generate a cytotoxic T-cell response in the patient, the therapy must then be administered by multiple intramuscular injections days to weeks apart. So, similar to the way prophylactic vaccines are designed to activate our adaptive immune system to recognize a potential pathogen, individualized neoantigen therapies are designed to stimulate a cytotoxic T-cell response against cancer cells already in our body. Thus, the goal of both prophylactic vaccines and individualized neoantigen therapies is to generate an adaptive immune response. Together, they form a class called immunogenic mRNA medicines. In the next lesson, we'll learn about mRNA medicines where the goal is to treat an existing disorder without generating an adaptive immune response. These therapeutics can be thought of as non-immunogenic mRNA medicines. Welcome back to the final lesson of our course, where we will learn about a third category of mRNA medicines currently in development, non-immunogenic mRNA medicines. In contrast to prophylactic vaccines and immunogenic mRNA therapies, where the goal is to elicit an adaptive immune response to either protect against an infectious agent or eliminate cancer, the goal of non-immunogenic mRNA therapeutics is to supply the instructions for a protein that can produce a specific therapeutic effect. Let's start this lesson by talking about protein replacement therapies for patients with inborn genetic errors. Please take a moment to review the learning objectives for this lesson. In the very first module, I showed you this metabolic chart which details all of the biochemical reactions happening inside of you right now to keep you alive and well. Many of these reactions are required for you to extract energy from food, while others enable your cells to create new building materials like fats and proteins. If you remember, each one of these reactions is mediated or catalyzed by a different protein, whose names appear here in blue beside each chemical reaction. These proteins that catalyze chemical reactions are called enzymes. It is estimated that around 1 in 1,000 newborns are born without the ability to make one of the metabolic enzymes shown on this chart. Such individuals suffer lifelong complications due to a small fault in their genetic algorithm. There are hundreds of different kinds of these so-called inborn metabolic diseases, dependent on which enzyme or biochemical pathway is affected. Because the vast majority of metabolic enzymes are intracellular, that is, they function inside cells, these disorders cannot be treated by supplying a protein therapeutic made outside the body. So for most of these disorders, no therapy other than simple symptom management has previously been possible. But because mRNAs can supply a patient with the instructions to make inside their own cells the exact enzyme they're missing, mRNA medicines known as protein replacement therapies are a promising new approach for the treatment of many of these conditions. Let's recall the inborn metabolic disease, glycogen storage disease type 1, or von Gerke disease, that we introduced in Module 1. Von Gerke disease is due to deficiency of a protein called glucose 6-phosphatase. This protein's job is to help release stored sugars so that you can maintain appropriate blood sugar levels when you're fasting. So, if you remember, children and adults who lack this protein can't fast. They must constantly consume small amounts of carbohydrates, including getting up every few hours during the night to eat raw cornstarch. And even if they're able to keep up this endless feeding schedule, these patients are plagued with many other lifelong medical complications. The organ that makes the most glucose 6-phosphatase is the liver. This is true for many metabolic enzymes. The liver is the largest internal organ in the human body. Because the liver is so large doses of therapeutic mRNAs are required to generate a therapeutic effect. So unlike vaccines, where a very small amount of mRNA, often as little as tens of micrograms, can elicit the desired response, enzyme replacement requires many milligrams of therapeutic mRNA. That is, over 1,000 times more. But this is still tiny compared to the grams of mRNA already present in the body. Further, whereas mRNA vaccines can be delivered intramuscularly or intradermally to stimulate an adaptive immune response, mRNA therapeutics for enzyme replacement must be delivered systemically, that is, intravenously, to ensure that it will reach the liver. Also, because mRNA is impermanent, lasting several days to a week at best, enzyme replacement using an mRNA therapeutic requires chronic repeat dosing. So, creating mRNA therapies for chronic conditions such as von Gerke disease require development of LNPs that are both efficiently taken up by the appropriate cell type in the liver and are suitable for chronic dosing. In 2018, Moderna scientists published a research article showing how lipid nanoparticles could be engineered to allow for repeat dosing via IV administration. This and other innovations enabled Moderna to take the lead in testing mRNA medicines for long-term treatment of genetic disorders caused by a lack of a specific metabolic enzyme. As of the recording of this course, mid-2024, the first enzyme replacement therapies using mRNA are showing promise in early-stage clinical trials. While not providing a cure, which is the realm of gene therapy and permanent genome editing, a major advantage of an mRNA medicine is that its dose can be adjusted according to each patient's needs. Also, if there are any undesirable side effects, the patient can either decrease their dose or simply stop taking the medicine. In addition to intracellular enzymes, another type of protein missing in some genetic disorders are proteins embedded in cell membranes. An example here is cystic fibrosis, or CF, the most common fatal genetic condition affecting people of European descent. Patients with CF produce thick, sticky mucus that can affect multiple organ systems, with the most problematic being the lungs. The problem in CF is an inability to maintain an appropriate balance of salt and water across cell membranes due to a fault in a protein called CFTR. Fortunately, many CF patients make a form of this protein whose function can be restored with a small molecule medicine. But about 10% of affected individuals make no CFTR protein at all. For this patient subgroup, a potential treatment is CFTR protein replacement via an inhalable mRNA therapeutic. Clinical trials are currently underway for these patients, with an mRNA medicine being delivered through an inhaler. LNP delivery vehicles for this medicine were specifically developed to remain intact during aerosolization, that is, the conversion of a liquid into a fine spray, and to be preferentially taken up by the cells lining the airways in the lung, whose job it is to make mucus more watery. Another potential use for mRNA therapeutics is in regenerative medicine, such as stimulation of new blood vessel growth, repair of damaged muscles, or scarless wound healing. Most regenerative pathways are initiated by one or more early signaling events mediated by proteins. These events kick off a cascade of subsequent events that eventually direct resident stem cells to divide and to differentiate into the desired tissue type. These so-called immediate early signaling events need to be highly localized and short-lived, characteristics that make it a perfect use case for local single-dose delivery of mRNA encoding the relevant signaling protein. Repeated dosing should not be required for these applications because the signaling protein is needed only to initiate the cascade. Examples currently being explored either preclinically or clinically include injection of an mRNA encoding VEGF into the heart muscle during open heart surgery to stimulate the growth of new cardiac blood vessels, or treatment of stress urinary incontinence by injection of an mRNA encoding a specific growth factor protein into damaged muscles around the urinary sphincter. While all regenerative mRNA medicines are still in early stage development, the preclinical and clinical data presented so far seem quite promising. Finally, another promising potential application for mRNA medicines is to stimulate the body to produce a specific secreted protein. In Module 2, we learned that proteins that work in the bloodstream can be used as medicines by injecting a pre-made protein. The examples we mentioned included insulin, clotting factors, and monoclonal antibodies. But mRNA medicines can enable the human body to be its own factory for making such proteins. In 2021, Moderna published a study showing that mRNA LNPs can be used to direct the human body to make functional monoclonal antibodies. In that case, it was antibodies capable of neutralizing chikungunya virus. Because the monoclonal antibody is made within hours of delivering the mRNA, this type of mRNA medicine can potentially be used to treat an already established viral infection. However, because antibodies have half-lives of only 10 to 21 days, the protection would be relatively short-lived. Thus, monoclonal antibodies cannot substitute for vaccines, which provide long-lived protection against future exposure to the relevant pathogen. Also in development are mRNA medicines encoding other secreted proteins, such as cytokines, hormones, and immune modulatory proteins, to treat disorders ranging from cancer to cardiovascular disease to autoimmune conditions. As of the recording of this course, no mRNA therapeutics have yet to complete the full clinical trial process necessary for regulatory approval. However, given their tremendous potential to address medical conditions where no good treatment currently exists, many companies and academic medical centers are now investing heavily in their research and development. Congratulations on completing the mRNAs as Medicines course. We hope this course has provided you with a comprehensive understanding of mRNA medicines, how they work, and their potential applications. The world of mRNA medicines is rapidly evolving with the potential to revolutionize healthcare. As we continue to explore and understand this fascinating field, we look forward to seeing the new discoveries and advancements that will undoubtedly emerge in the future. Thank you for joining us on this exciting journey, and we hope that you will continue to learn about and explore this revolutionary field of medicine.